So, I think I'm online again. Hi, hello, and welcome again. Microbehunter here. Welcome to another Saturday live stream, Saturday microscopy live stream. I'm happy that you could join me again today. Today I'm going to do again a little bit of lab work. It's a requested video. Basically, I'm going to do a so-called a gram stain. Gram stain, let me quickly check here. I need to get everything sorted out here. A gram stain is a very important stain in microbiology. And I'm going to give you a live demonstration of this gram stain today. And I'm not saying that this is something that every hobby microscopist actually has to do. It's not necessary, but I think it's still interesting, or I don't know if it's important, but in any case, I hope it's in any case interesting to know how it works. So basically, I'm doing the gram stain for you so that you don't have to do it, to put it that way. Now, I'm seeing that many of you have joined in again and have said hello already here in the chat. Yes, please feel free to comment in the chat, and if you have any questions, then please indicate this with at Microbehunter or at Oliver, and then after approximately 1 hour, I will try to answer the off-topic questions. But if there are some on-topic questions, of course, as always, I would like to address them right away. I'm going to spend the next 2 or 3 minutes simply going through the chat, saying hello to give more and more people time to actually join into the live stream. So there is a hello from Athens in Greece, from Germany, from Belgium, from Western England, the United States, Netherlands, France, England, from Finland. Video and sound is good. Thank you very much. Okay, from the Netherlands again, from Germany, from Lebanon, from Austria, from England, from the Philippines, from Australia, from UK, from Wales in Austria, from Kentucky, United States, Netherlands. Okay. And how are the slime molds doing? Marcel wants to know, and I will tell you the slime molds are now completely dried up on a piece of filter paper, and I put them away into a little plastic bag. Okay, so hello from Ohio, United States, Southern Ireland, and you know what? I'm just gonna get started right now with the lab work. I'm doing the first thing is a little bit of a theoretical background, and then I'm going to interrupt myself again, and I'm going to go back into the chat later in a couple of minutes. So what am I going to do? I'm going to, before I explain to you all of the materials and everything that we need here, I'm going to quickly show you now a live view under the microscope to actually show you what a gram staining of bacteria actually is. Okay, so let's have a look at the microscope view, and I prepared today in the morning, my time, today in the morning already a slide, with bacteria. Okay, what you see over here is these are bacteria, and let me quickly. Do I have an arrow somewhere? Here it is, the arrow. Okay. So these are basically all of these dots that you see here are bacteria, and you see that they are basically stained red. Okay, so these are so-called gram-negative bacteria because they are staining red. And I'm now going to also show you a bacteria that stain gram-positive. I'm moving this over. Look, and these bacteria here, they look much darker and they stain gram-positive. Okay, so in other words, the staining technique, the gram staining technique that I'm showing you here is allows you to distinguish between so-called gram-positive and gram-negative bacteria. The gram staining technique was developed by Hans Christian Gram. He was a bacteriologist in the 19th century, and this gram staining technique was very important and still is used today. I have myself used it during my university time. I can talk a little bit about that as well. Okay, I'm also going to talk a little bit about biology, how the gram-positive and gram-negative cells, how they're actually different from each other. Okay, so again, these are the gram-positive bacteria. You see that they stain dark purple, dark blue, almost black. Here, very dense over here, right? And over here on the other side, I move up, wrong direction. Here, I have to go over here. You see, yeah, they are red, right? So they're gram-negative. Yeah. That's basically the staining technique, and I'm going to now run you through the protocol, so to say, the steps that I'm going to do so that you understand a little bit of what I'm actually doing here, right? So let's have the desk view again, and then, yeah, so here we go. So here are the steps. Yeah, basically that's Hans Christian Gram. I got his picture here. So he's from the 19th century, and yeah, he developed this technique, and it's of course not only of historical relevancy, but it's still used in bacteriology these days. Okay, you need a couple of stains here. You need crystal violet stain, and then you need an iodine. You need to decolorize or wash everything, and then you need to counterstain using safranin solution. Okay, and what I'm going to do is I'm going to explain to you first for the next couple of minutes of actually what happens, because I think that's also important. Okay, after all, we're doing this live stream here because you want to learn something. I want to learn something as well, because I had to also experiment around a little bit on getting this optimized for the live stream. But essentially, you need crystal violet, iodine, and safranin, and you also need alcohol here. Okay, for washing. And basically, I've got everything set up over here in my limited desk space that I have, and of course, I need some bacteria, which I've also got over here. Okay, so I'm going to quickly explain to you what actually happens here. Okay, so this is going to be a 5-minute biology lesson, which I think still is important because we want to understand what happens here. Okay, so and then after this short 5-minute biology lesson, I'm going to explain to you what happens. So essentially, there are two different types of from the cell structure now. Okay, two different types of bacteria around. The one is called the gram-positive. I abbreviate this with GR-positive. They stain blue-black. Blue-black. You've seen them, just before. And the other ones are called gram-negatives. I abbreviate this with GR-, and these stain red. The question is now, how are they different from the cell structure? And the difference has to do with the thickness of the cell wall and the number of cell membranes. So in a gram-positive bacterium, very simple sketch. I've got the cell membrane over here, okay, or plasma membrane, and on the outside here, very thick. I know this is a very bad drawing. All of this here is the cell wall. Okay, so it has a thick cell wall. Cell wall on the outside, right? So that is the gram-positive bacteria. Gram-negative bacteria, on the other hand, they have two membranes. One, then a second one here, two membranes, and in between the outer and the inner membrane, you have a thin cell wall. Okay? Yeah, so you have a thin cell wall in between. So cell wall, which is thin, and the cell wall over here is thick. Okay, and because of this structural difference, they stain differently, and I'm going to explain to you also how this actually works. Okay, so basically one membrane here with a thick wall on the outside, and over here in the gram-negative we have two membranes with a thin wall between the two membranes. Okay, and what happens now is the following. What I'm doing in the first step over here is I'm going to, after I have heat fixed them, and I've prepared everything, air dried them, heat fix them. This is going to take a couple of minutes. I'm going to stain them with crystal violet. This one over here, right? Some of you already know it because I've been using this stain before, and it's a very, very strong and powerful stain. And do you see those blue spots over here? Yeah, that's the crystal violet. It's so strong that I had problems even getting it out here of the table surface. Okay, crystal violet is a very strong stain. Okay, and then I'm going to do the following. I'm going to add iodine to it. And what happens? So basically the crystal violet will actually go into the cell, gram-positive or gram-negative, and it will stain them blue. And then when I add the iodine solution, which is this one over here, okay, then what's going to happen is it's going to form a crystal violet iodine complex. So in other words, it's going to react with the iodine with the crystal violet, and it's going to make it's a chemical reaction which stays inside of the cell. Okay, so basically it becomes less soluble, and it will start to bind the crystal violet inside the cell. And then the next step is the following, is the decolorization or the washing stage. And what happens now is the following, that in the case of the gram-negative the gram-negative ones, what will happen is that the alcohol is able to remove the crystal violet again. Yeah? So the stain is going to be removed from the gram-negative ones, and those basically will start to lose the blue color again. Yeah. So what we have then is we have one that stays black, blue, and the other one which loses the color because of the alcohol, and this has to do something with the membrane. Okay. And then we're going to counter stain the whole thing with safranin, which is red, and this basically makes the gram-negative cells red so that we're able to see them. Right? So essentially, in summary, it's actually this decolorization or washing step which determines whether it's going to stay blue or not. Okay, because the gram-positive ones are able to retain the color. Right? And then the safranin, which is this one over here, the red one, is the counter stain so that we're also able to see those that have lost the color again. Right? So this is a little bit the theory behind it. And the nice thing about the gram stain is that actually you are able to see the difference, so to say, in cell structure over the stain. Now, Mr. Gram back in the 19th century did not know about these differences. Yeah? And the reason why he came up with the stain is actually had a medical reason, because he observed animal tissues that were infected with bacteria. But the problem is that I think I read that he also looked at lung cross sections, for example, where there were bacteria growing inside the lung. But the problem is if you take a lung or any other body tissue with bacteria and put it under the microscope, you're not able to see the bacteria because there are so many body cells that will actually cover up the bacteria. Right, so from the proportion, there's simply many more body cells, and the few bacteria that you find there are generally not visible. So he tried to develop a staining method, the gram staining, which basically allows doctors to contrast the bacteria and see them better in comparison to the remaining body tissue. Right, and that's basically how it was developed. Right, and this was already the theory part. And what I'm going to do now is I'm going to interrupt rather. I'm going to have a little break with my little monologue here, simply to read again some of your comments here. Okay, so hello from Florida, from Poland. Okay, I'm going to skip over all of those parts that are not directed to me. Okay, are there risks involved in performing the gram staining? No, not really, because the substances used are not really dangerous as such. I mean, you always, I'm not using methanol, that's important. The stains are standard stains and there should not be any risk. I will tell you what the risk is. It is because I'll be heat fixing the cells. This means I'm going to be working with a fire, of course, and that might be a risk. Okay. I can see how Oliver built up to this over the last months. Crystal WhatsApp, yes. Paul is commenting, yes, over the last couple of months, I kind of bought all of the stains together. Okay. And yes, there is an off-topic question about the so-called the conjugate distance of a microscope objectives. I'm going to skip over those parts and I'm going to answer all of those off-topic questions later on. Okay. So now I'm going to start, and now we have to be a little bit patient, okay? Because we have to do some air drying of the bacterial suspension as well. So what else do I have here? You already know this little friend here. That's my coffee warmer, which I'm going to be using, okay? To air dry the cells. And now I'm going to show you some of the Petri dishes that I have. And honestly, I had some, actually I nearly decided not to do this live stream today on this topic because those Petri dishes are an embarrassment. Okay, so just a big disclaimer. I say an embarrassment because actually I'm a trained microbiologist, but the Petri dishes are pretty much the worst ones. The worst ones ever. I will explain why. Okay, at home I don't have a full laboratory setup, so I try to improvise. Okay, and I'm going to show you the Petri dishes here. And I wanted to throw them out, but then I said, well, actually, maybe it's an advantage because now I can do some gram staining. So I'm going to show you the Petri dishes that I made. And honestly, this is an embarrassment. Okay. Look at this here. What do you see here? You see here, I made the growth medium. I did not have an autoclave. I just boiled it over the plate. It wasn't enough. So what you've got here, those dots that you see here, these are bacterial colonies. And the thing that you see over here, that is because I had this condensed water running over it and distributing the bacteria. These colonies should not be there. Why should they not be there? Because these are contaminants that fell in from the air, right? So I was not working sterile. Right? That's why I'm saying it's an embarrassment. This is a disaster, okay? This is a no-go, right? And over here, this one here, another plate, I tried to grow some E. coli bacteria. I've got E. coli in capsules, but the problem is that because I did not boil the medium long enough, the agar medium did not get solid. So basically I had problems streaking out the cells here, yeah, because the agar was so soft. So it was a disaster. Okay, but at least these things that you see here are gram-negative E. coli bacteria, so which I'll be using. So in that sense, it's still workable. Okay, and you see over here as well. Yeah, and those tiny colonies that you see here, those tiny dots here in the background, they're not on the surface. They are actually inside the growth medium because it didn't disinfect properly, okay? So I will quickly explain to you, those of you who are completely new to microbiology, I want to explain to you what a bacterial colony is. So what I've got here is, in this Petri dish, I've got a nutrient medium which contains nutrients like yeast extract, and also a so-called agar-agar, which makes it like a gel-like solid. And when there are bacteria now on the surface, a single bacterium, you're not able to see it, obviously, because it's microscopically small. But this bacterium will now start to divide from 1, 2, 4, doubling 8, doubling 16, 32. At the beginning, not a lot happens, but it's exponential growth. This basically means that after a couple of hours, you have billions of bacteria in one colony. So each one of these dots that you see here, right, contains billions of bacteria, but comes from one original bacterium, right? So by counting those colonies, you can actually determine how many bacteria were present in the first place. Right? And again, here on the side here, that is because there was some water droplets running over it because I messed up the medium, right? And it kind of spread the bacteria as well. So that's why these are very bad cases. But at least, yeah, I have now some bacteria. There's also some water condensed here. So I now need to say the following. Look, folks. Which bacteria are these? I have no idea. I don't know what they are. They're contaminants from the air, or maybe they were already in the yeast extract a little bit. I don't know what they are, right? So I have to be careful, right? And that's simply something where I just want to warn you. I've been doing this a lot at university back in the day. Those here are mostly E. coli bacteria. I say mostly because they're also contaminants over here. But you don't want to mess around with bacteria, especially those where you don't know what they are. I have absolutely no problem with yogurt bacteria or things like that. And these E. coli bacteria actually also come from capsules that can be swallowed to regenerate the digestive system in case you have some kind of digestive issues. But those contaminants, I have no idea what they are, right? So we got to be careful. Okay, so I'm just saying this because I've seen on YouTube, YouTube channels where some people just basically make those agar medium, they make fingerprints to see how many bacteria are. And I say, no, you don't want to do that. You don't want to grow your own bacteria because these are, I don't know, Staphylococcus maybe? I have no idea, right? You want to be careful, especially if they're able to grow on the human body, right? So just saying, yeah. So this will be our source for bacteria. And what I'm going to do is I'm going to take where was this one? These one here, because these are E. coli bacteria. Again, very bad because I did not spread them properly because of the I have over here the favorite tool of a microbiologist. This is how do you call this? An inoculation loop. For those of you who speak German, an Impföse. Okay, it's an inoculation loop which allows me to pick up colonies here. And this is basically what I'll do. And normally, every time when you work in a microbiology lab, you need a gas burner. I've got one over here. And I'm not going to use this because it's too dangerous, right? Because I have it basically then here, and then I'm going to break my camera, which is right on top of it, right? So what I'm going to be using is I'll be using a little alcohol burner here, which is, because I'll be disinfecting this. So what I'm going to be doing is the following. Number one, I will be preparing, okay, I'll be preparing, over here a microscope slide. And I'm going to, first of all, put a small drop of water basically on here. And you know what, I'm just going to use, it's just, theoretically, I could actually put both samples here. Yeah, maybe let's do that. Let's just put two different ones over here. So a little bit of water, okay, here, a little bit of water over here, okay. I will now turn on the lamp, making sure that maybe I'm doing it like this. How am I going to do this? Or like this? Yeah, let's move it away a little bit so that because of the heat of the lamp. Yeah, that's fine. And here we go. And the first thing that you want to do is you want to basically hold it into the flame until it glows. This basically kills off any bacteria that are possibly on the loop here. Yeah, and I'm going to now do the following. I'm going to now take this here, open it, cool the wire. It makes a little sound, and I'm going to now grab a little bit of this colony over here. Okay, there's not very much on here. Okay, and I'm going to try to suspend it now. Yeah, in here, and I'm just holding it. Yeah, there is something in here. Not a lot. Okay, not a lot. And when you're finished with that, you just basically, go back here again. Yeah, this is very soft. See, I was not even able to properly pick it up, right? Because it was so soft. Yeah? Maybe add a little bit more. You see, this is actually the whole colony. I couldn't even properly pick it up. Okay. So let me hold it over. Yep, there is. Sometimes if you don't see it, then you want to hold it over a dark surface. Yeah. So that's number one, an unknown one. And over here, I'm going to do now the same thing over here with with the E. coli. However, it also contains a lot of contaminants, which is okay. For our purposes, it's just a demonstration here. See, I have a real problem picking up cells here. The medium is so bad that essentially, there seem to be also cells present right now. What we might want to do is we might quickly put it under the microscope simply for a quick check. Okay, so that's basically finished. This one is closed again. Okay, let me quickly, I'm going to quickly put it under the microscope to have a look without cover glass. Okay. So let's try this, and then while I'm waiting for it to dry, that's still the other one. Let's put this one in here. You don't need a lot of cells, but there is plenty of stuff here. Yeah, all of those tiny dots, at this magnification. Yeah, I think you get it. These are all cells, and I'm using now my 20 times objective. So this one over here seems to have a little bit fewer cells. Still enough. Okay. Now they're pretty small now because I'm using a very low magnification because I don't have a cover glass, and I don't want the objective to touch the liquid, right? So this was just a very quick check if I have anything on here at all. I do have it. So what I'm going to do now is I'm going to take it again, right? And I'm going to now air dry it where am I going to air dry it? Because I'm very impatient, I'm going to just use my coffee warmer here to air dry it. Yeah, and we're just going to wait a few minutes until it's completely air dried. Okay. In the meantime, I can do a little bit of cleanup work here. Okay, and yeah, and then we're going to basically continue the actual heat fixing and staining process. So I'm going to read now about my first microscope. The reason that is the plan and semi-plan objectives. Okay, that's a comment here. Can you stain different gram bacteria at the same time on the same slide? Yes, this is actually what I have done right now, simply because I was lazy. Okay, no, not only because I was lazy to switch slides, but simply because it's later on easier then to observe it under the microscope, because I just move over and then I can actually see both of them next to each other. Okay. So high is the multiplying or rate time sometimes used as a defining criterion for identifying groups of bacteria. Okay, that's an interesting question. Which the question is do basically the speed of bacterial reproduction, can this be used as an identification criterion? And I would say probably not, because the speed of reproduction not only depends on the bacterium, but also on the nutrients available. So if the nutrients and the conditions are right and the temperature is right, then the speed, of course, is faster. Now you're going to say, well, we could theoretically standardize that. So if under standard conditions, could that not be some kind of an indicator? And I think still not, because it is a single criterion. Now, this basically means highly related bacteria could maybe reproduce at different rates. And just because two bacteria reproduce at the same rate does not necessarily mean that they're also similar, right? And maybe two different bacteria can reproduce in the same rate. The example that I'm always using is just as an analogy now, just because two cars have the same color does not mean that the two cars are the same brand, right? Or the same make, right? So you have to look at more characteristic, typical. Fast growing bacteria can divide, for example, every 20 minutes. There seems to be a lower limit, because the limiting factor in bacterial reproduction is the speed at which the DNA can be copied. Right, and generally fast-growing laboratory bacteria, which are optimized, basically lab E. coli bacteria, can reproduce divide once every 20 minutes. But this seems to be somewhat the lower limit, because it simply takes time for the DNA to copy, and that's basically the bottleneck, so to say. Oh, I'm digressing. Okay, but it's an interesting question. Are we going BSL2? What is BSL2? This is basically biohazard safety level 2. Strictly speaking, every time when you are rotting food in your refrigerator, you are already in biohazard level safety level 2. So when you have unknown, if food is spoiling, you don't know which bacteria are growing there, you're already, strictly speaking, per definition, you're dealing with unknown bacteria. Right? So interesting. We use the cover slip to spread it out. Then the heat fix. I actually don't do that. Okay. I will show you what I'm doing here. You see that this one already is dry. This one over here not yet. You can actually now see that the color has changed. It has turned white, and this is actually a nice sign that essentially the slide has dried. Okay, we can, and now then I need, when this is completely dry, it has to be heat fixed. It's important that you do not heat fix it before it's dry. That's important, otherwise the cells are going to pop open and it's not going to work. Now, I did a lot of gram staining back in the day when I was still at university, and not for identification purposes alone. We did our DNA analysis and other chemical tests to do that, but as a ways to characterize it. And I found out that gram staining was actually a thing I did not like to do a lot, because in order to get highly reproducible results, you had to standardize the staining a lot, right? Bacteria that were older did not always stain the same way than fresh bacteria. So there was even a change there. So we found out that gram staining, you don't just stain it, but there are lots of things, parameters that you have to standardize. That also the time of washing and the staining time and everything. All of these things I'm not going to do now, okay? But I just want to tell you that if you want to do this in a laboratory grade way, then you cannot just take any old plate and do gram staining, but actually you have to determine the time and the age of the bacteria of the growth stage, and then you have to and then you do the staining, right? Because old bacteria might stain differently than young bacteria, and so on. Yeah. So this is a little bit the thing here. So this one now basically seems to be completely dry, and this means that I can now do the heat fixing, and the heat fixing is important. First of all, what does the heat fixing do? I will now pull the slide specimen on top away from the flame. I will pull it through the gas to the, in this case, the alcohol burner a few times, because this will not only kill the cells, but it will also stick the cells to the slide. It's like how do you call it? Like an egg in a frying pan that sticks, yeah, or meat that you fry without oil. It will stick to the frying pan. And this is what we're doing here as well. It's important that the cells stick to the glass slide. Otherwise, they're going to be washed away during the staining process, and that's not what we want. Okay? So heat fixing is absolutely central, otherwise they're going to be washed away. How hot should it be? There is a general rule of thumb that says after the heat fixing, you should be barely able to hold it in your hand. You should be barely able to hold it in your hand without burning yourself. And I just realized that essentially it was 55 degrees, it was already kind of warm. So I hope that this actually didn't hurt the cells either. Okay, so we're gonna do that. So I will basically pull it through the flame here for a couple of seconds. I will test it. It's hot, so that's enough. Okay. I don't wanna burn the cells, obviously. Yeah. So I have to wait now until it cools a little bit. So I have to put it on a cool. If I put it on the plastic surface over here, then it's going to melt. So usually, you can put it on a glass Petri dish maybe. Yeah. Or maybe I'm just going to put it on another. I don't know. I'm just going to keep it here, and I'm going to wait until it cools. It's still quite hot. And what I'm going to do is I'm going to do all of the staining on this tissue paper here. So I'm just going to put it here because otherwise, I might get some of the stain here on the surface. So my only concern is now is I hope that this 55 degrees here that I had was not too hot, because if it's too hot, then the cells will actually pop open, right? And I don't want that. But what we can do now is just before we do the actual staining, why not have a look under the microscope in any case? Yeah, why not have a look? It's still kind of warm. It's kind of surprising how long the glass slide actually is able to keep the temperature. So that's fine. So let's quickly have a look here. I mean, we already looked at it before, but now let's have another look, just for the fun of it. Where is the scope, the microscope view? Here we go. And so that's basically how it looks like. And over here, these are the other ones. You can actually see that as the slide dried, the cells were sticking together in this kind of pattern. But now, because there is no liquid here, I can easily go up with the magnification as well. Okay, all those cells are kind of sticking together. Okay. So that's all. By the way, I removed the DIC filters and so on. So that's just regular bright field. Yeah. It's just regular bright field. And I don't know if both of them are gram positive or gram negative. So we're going to see. So we have basically, it's a little bit a question of luck. You see how the cells are sticking together? They're barely distinguishable. Yeah? So doesn't matter. I hope that they're different, or at least that they're a mix. Okay. And we're now going to actually start the staining process. And this takes now a little bit of, patience. And we're going to get ready. And the first thing that we have to do is crystal violet. Stain for 1 minute and then rinse with water. Okay, we're going to do that. Okay, so where's my crystal violet stain? Okay, so this is a very strong stain. And I'm going to put it directly over here. I have to see where is the here and over here. And now you know I've got these toothpicks here, because I'm just going to use those toothpicks to spread carefully without touching it over it, so that all of them essentially are carefully covered. Okay? So that's basically, yeah, step number one. Okay, and as I'm waiting, I'm going to read again here. Before DNA analysis was commonplace, how did they identify bacteria? Chemical procedures that took forever. Yeah, that is. I think that's actually. You know what? Thank you for the comment, because tomorrow I want to actually put out another podcast, and so maybe I'm just going to talk about that as well, very briefly. What you can do is, no, I'm going to start in a more basic. It depends a lot for identifying bacteria. Up to what level do you want to identify it? But in principle, the following things are possible. Number one, DNA analysis. I think that's the best way, most expensive. Yeah. Number two, what chemicals are the bacteria able to use as a food source, and what end products do the bacteria produce? So basically, do the bacteria produce certain acids when they're growing or whatever, right? So what end products do they produce? And the third thing is to do a so-called a biochemical analysis of the components. This was actually the topic of my diploma thesis at university. I had to grow bacteria, and I did analytical chemistry on the parts of a cell. Basically, which lipids are present, in the cell membrane, and so on. So there are basically, you use analytical chemistry to do that. It's called it's called chemotaxonomy, and basically you look at the chemical composition, and generally the microscope was not used so much, because the microscope itself was used for quality checks, but does not really produce a lot of information that allows you to really identify the bacteria. Okay, so step one was the staining. Then what do I have to do? It says here, rinse with water. I'm going to show you now how I'm going to do that. So the crystal violet I can put away already, and I'm going to rinse it with water. And yeah, here on my table, I don't have enough space. It's of course much nicer to have a properly equipped lab. But look what I'm doing here. I'm taking this now. Over here I've got water. You don't put the water directly on the bacteria, but above it, okay? And you simply allow it to carefully run down, okay? Okay, so that's basically the washing stage. What do you notice? What do you notice? Look, yeah, they've stained blue. Okay, you can actually see it now quite nicely. Yeah, so by the way, just for your info, side info, the blueness, so to say, or the blueness of the stain can also be used as a measurement for density, for bacterial density. So the darker it is, the more cells are present. Okay, so there are actually devices that are able to do this measurement. So crystal violet is not only used to make cells visible, but actually can also be used to quantify and then to measure out how many cells are actually present in the first place. So that's number one. Okay, so step one is complete. Now I have to add iodine. Apply for 1 minute. It forms a crystal violet iodine complex, and then I have to rinse it. So I have to now add some iodine to it. So where's the iodine? Over here. Same story. I'm going to use again a fresh toothpick in this case here. So you want to be careful with the iodine as well. It's also a very strong one. Yeah. So here it's iodine. And because I like to not use too much of this, that's why I just put a drop on here, and I again use carefully a toothpick to spread it across. I'm just doing this so that I don't have to use too much of this iodine. Look how the iodine reacts with the cellulose here. And I have to wait a minute again. Yeah. So I'm reading again. Isn't RNA sequencing similar to DNA analysis? The point is the following. The RNA of a cell changes all the time, depending on which regions of the DNA are being transcribed. Ooh, this was now too fancy. In other words, the DNA stays the same in a cell and the RNA is different. Okay, basically the RNA are just copies of the DNA, but the cell will actually decide which sections of the DNA to copy. So I would say that RNA sequencing, is probably not used so much for cellular for identification. You use DNA sequencing. Yeah? Maybe there are some uses for this, but I've always been only doing DNA sequencing. Yeah. Before adding iodine, how does it look? Oh, I think I just showed you. It was just blue. Yeah. Okay. So that's one. This was basically now the iodine treatment, and now basically the iodine reacted with the crystal violet inside the cells and basically formed this crystal violet iodine complex. So let's next step is, what does it say? Always follow the instructions. Rinse, of course, right? And you already know how this works. Yeah, you go like this. Now you carefully again apply this here on top, and here we go. And you actually can see now that that's actually also quite interesting. It's not quite as blue anymore. It has become a little bit darker, right? Almost like dark gray or black. Okay. So now we've got this here. Next step is critical. Brief alcohol wash and rinse immediately. So this means now I have to be a little bit careful. I have to now wash it with alcohol, and the gram-positive cells will retain the crystal violet iodine complex, and the gram-negative cells will lose the color. Okay, so that's the idea here. So how am I going to do that? And because I don't want to use too much of this alcohol here, I decided to do it like this. I'm just going to put in some alcohol in here. Okay, and I'm going to use a pipette now, simply to save a little bit the alcohol. And look what's going to happen if I now add some alcohol over here and also over here. Okay, you can actually see that it turns a little bit. Yeah, it's here. It kind of turns blue again. You can actually see how how it's being washed out. Okay. And you have to keep on doing this until no more stain is washed out. Okay. But I think this seems to be okay. Sometimes okay. Yeah, it's a little bit purple still. Okay. So I'm going to maybe last time. Okay, it takes patience. I mean, honestly, look, just by looking at it already, we can actually see, and I hope that this is going to be confirmed, that this one seems to still be significantly darker than this one over here, right? This one over here, actually, I can still barely, I don't know if you're able to see it, I can barely see something here, yeah? So we're going to, a little bit you're able to see it. So, and the next step would then be, there's still something coming out here a little bit. So, I think that should be enough now, okay? The next step now is the following. It says here, and then rinse immediately again. So I'm going to remove the alcohol again with a little bit of water. Okay, here we go. So, okay, so I'm now going to read again some of the comments. Okay. I see Jesse's just saying it would be nice to see it under the scope after each step. I get it. That would be actually a good that's actually a good point. I didn't think of that. But you know what? What time is it now? Let's try, why not? Now that we know how this works, then maybe I'm just going to try it again yeah, just showing it with each step. But you know what? I'll just put it also under the microscope just to see if there is now actually a difference. Let me see. Scope. Let's have a look here. So. Okay. Okay, that's how it looks here. And over here. That's how this one looks over here. We still have, it's not, yeah. I'll be using, yeah, let's have a look here. Yeah. Let's see how it looks like after the staining with, let's have a look at how it looks like after the staining with saffron in. This is desk view. So, and last step is counter stain for 1 minute, rinse and blot dry. Okay, so let's try this here. Here and here. Let's spread it again a little bit carefully. Okay. And then, yeah, I wonder how this will actually look like. Okay. If you're pressed for time, you can always do this as a short or a mini video later. Yes, I have definitely intend to do that because there have already been several requests for gram staining demonstrations, and I think a full hour just in that detail might not be necessary in any case. Yeah, so I'm indeed, and then probably I want to really use. That's my intention to use maybe yogurt bacteria, because yogurt bacteria are also gram positive. Okay, and then you've got E. coli and yogurt bacteria, you've got a gram positive and a gram negative bacterium, and then you can actually nicely compare it side by side. Yeah. Okay, so a mixed bacterial group will show red and blue bacteria. That's correct. And thus give some info of what it is. Well, yes, you can only say one of them is gram positive, the other one is gram negative. You can kind of go into the direction a little bit, but it won't allow you to actually specify in detail which one it is, right? But at least you can separate out the groups. I mean, for example, I'll give you an example. It's used for quality check. If you are working in microbiology and then you're supposed to work with E. coli all the time, and then all of a sudden you've got a gram positive bacterium on your petri dish, you know that you're working with a contaminant. Yeah, so that's so a little bit the possibility to do some quality checks. So I think that's enough now, and let's have a look here. I'm going to rinse this again. Okay, and here we go, and then we're gonna see at least the E. coli one. If it were a pure culture, it should be completely red. The other one, I don't know. It also kind of looks red now. So maybe both of them were gram negative. Yeah, but in any case, I'm going to now do the following. I want to use now oil immersion. Okay, so that's now the next thing. How am I going to do that? It has to be completely dry for oil immersion microscopy. So what I'm going to do is I'm going to put it again on my coffee warmer here for a minute or so, simply to evaporate the water. Okay, because yeah, I want to go up to the maximum magnification. So yeah, okay, there are some, yeah, Beerman funnel. Okay, contaminant warning. Okay, I'm just going through the comments here. Yeah. Yeah, I think what I'm going to do is I'm going to do a separate video of this later on as well. What time actually is it? Okay, we've got 15 minutes already. Yeah, just saying, my suggestion is, if you are interested also in observing bacteria and so on, don't grow them on petri dishes because of contaminants. Just go out and get yourself some yogurt. And put some yogurt, especially the liquid part, not the solid part, but sometimes there's this liquid, watery thing in a yogurt. And if you observe that, you're going to be, you're able to find a lot of bacteria there, as well, yogurt bacteria. They're gram positive. That's perfectly fine. They're safe, but you don't want to work generally for hobby purposes with bacteria that you don't know. Yeah. So that's, yeah. They're both gram negative. Could be. I mean, at least the E. coli one should be gram negative. Yeah, but then I'm going to show you over here. I have over here. Where is it? No, it's not this one over here. Is it this one? Ah, yeah. From today in the morning, I've got another one prepared already with some immersion oil, which contains gram positive and gram negative ones. So this one now is dry. Okay, that's good. So, and I'm going to now just put it under the microscope, and we're going to look at it without oil immersion first. I mean, you already know that I'm not a fan of oil immersion, but occasionally it is necessary. Let's remove the arrow as well. Ooh, it seems to be gram negative. Oh, that's just a second. That's the wrong arrow. Arrow gone. Arrow gone. So, ooh, this seems to be. Oh, also the first one. That's definitely red, right? Okay, I'm gonna show you then. That's why I made the other slide as well. Yeah, and the other one over here is also red. Okay, let's go up 40 times. So both of them seem to be gram negative. Maybe if we're lucky, we find some. It's not a pure culture, and there's some contaminants. No, that's definitely red. Okay, so 60 times. Yeah, usually when they're clustered together, then yeah, the color is more evident. But you do see that the shape is different, right? Of the bacteria. So they seem, both of them. I mean, that's, yeah. Yeah, both of them are gram negative, unless we're able to find the occasional contaminant, which I cannot find here. Dental plaque, that's also an idea. Saliva contains a lot of, yeah. So, and now let's do the following. Let's do a little bit of oil immersion microscopy, and I do have another slide here as well. And for oil immersion microscopy, I'm going to now do the following, and you might want to check a little bit of what I'm doing here at the top, right? Without immersion oil, okay, that's without immersion oil. I try to get the focus, and it's kind of blurry, right? And then what I'm doing is you cannot see that, but my microscope has this focus lock lever. Okay, so I'm now setting it, so I basically, it will now remember the position. Look what happens. Look what happens. I'm now using my coarse focus knob to lower the stage, and now I'm moving it up again all the way, and it stops here already. I cannot crash the stage into the objective because it remembered the topmost position. Okay, yeah. So I'm going to now do the following. I'm going to lower this here. I'm going to add now a drop of immersion oil. You just watch what I'm doing here at the top. Okay, here's my immersion oil. A drop goes on here. A drop also goes on the other one. And now I'm going to rotate it up again until the topmost position, which remembered. Boom. You see it lost a little bit in the focus, and here we go. Okay, you might want to move it a little back and forth, and you see that actually the image quality is much more clear than before. Number one. And let's have a look. I wonder if you are able to see now much better that indeed we don't have a pure culture here, because you're able to see, and that's actually quite nice. Arrow, 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 arrow. You see there are a whole bunch of tiny bacteria here, and some of them here in between here are staining much darker, right? They also have a different shape. So these are the gram positive ones, right? And here on the outside, those tiny ones here are gram negative. So it's actually quite a nice. I'm quite happy that I, because of this, the contaminants of my plates, yeah, you can actually see that there's a mix of different bacteria, and you see that they don't only stain different. Here, that's a nice one. You can actually see that they also stain differently and also have a different shape. Yeah. Yeah, so yeah, See the red bacteria here, the tiny ones on the outside here, and here in between the dark ones here are the gram-positive ones. Would you use oil immersion at 100x and phase contrast if stained too? The problem, no, if I stain it, I do not want to use phase contrast because phase contrast is going to make it more grayish, okay, and you won't be able to see the colors. Yeah. So Staphylococca and just cocci, I don't know what they are. Actually, this was the place where I added some E. coli bacteria. So these are gram-negative ones, and because of so many contaminants, yeah, on the plate, yeah, these are the other ones. So I'm going to now move over to the other side. Okay, I hope that the oil film doesn't rip. Okay. Ah, yeah. And they are also gram-negative, but you see that the cells are a little bit bigger. Okay. Yeah, they're also rods. And the reason why they are clustered together like this is because as the water dried out, they started to cluster together. But you see actually over here, yeah, here's an individual cell. Yeah. So, but I kind of like the other one a lot more because it actually shows a nice mix. Yeah, and of course, the reason why this one does not contain any contaminants is, remember, I took it from one colony, right? So, of course, we expect all of them to be the same. This was one of these big colonies, while the other one I kind of tried to scratch off somewhere in the center, and here again. Very nice. You know what? I'm going to show you also the other one that I made today in the morning. Okay, and here it is. You can also check what I'm doing here at the top. And I have to lower this here again. I'm going to lower it again. I'm going to now move over here. And I hope I still have it. Now I'm going to add a little bit more oil maybe. I'm going to add a little bit more oil. And I'm going to raise it again. Let's see if I'm able to find the focus. Stop. Yeah, and that's the one that I made today in the morning. Yeah. You can also see that there is this mix between gram-positive and gram-negative. Here, mostly gram-positive. That's actually, by the way, remember the video that I quickly showed you at the beginning, or the picture at the beginning of this live stream? Yeah, this was actually the sample over here. Yeah. Yeah, so that's basically, yeah, a demonstration of gram stain. Yeah. So I'm going to quickly go through the comments again. Okay, there are no questions for me so far. Okay. Can I use this method to analyze my saliva while I have a cold? You can do that, but I will be honest with you, when you have a cold, then this is a virus infection and not a bacterial infection, okay? So you can essentially, you have so many bacteria growing in your mouth anyway, it doesn't really matter whether you have a cold or not. So yeah, that's actually also a possibility. I don't know, how much time do I have? Yeah. I don't know if I'm able. The problem with saliva is there's so many cells from your mouth also in there. So I don't really know if it's a good how do you say specimen or not. But you know what? That's always the risk of a demonstration. You never know if it's actually going to work out or not. So if you actually already want to know about saliva, you couldn't see this now because the desk. Here it is. Here's my saliva. Unfortunately it makes foam, and and I'm going to put it on here again, and I will tell you the following: there will be probably a lot of body cells from my epithelium on here as well. Yeah, you can already see that it's starting to dry up. Would epithelial cells color and bacteria differently? I don't know. Honestly, that's a good one. I can imagine that maybe the body cells that they will not retain the stain because they don't have a cell wall. Yeah, I would guess. I don't know. But in any case, Graham, the guy, he tried to actually use the staining technique to make bacteria specifically visible. So I can imagine that maybe the body cells that they do not stain gram-positive. Yeah. If I have an infection with a cough and I'm throwing up mucus, what then? Well, most likely, if you have a cough, of course, it's possible that you have got bacteria also, yeah, that cause the cough. But if you've got the common cold is actually caused by a virus called the rhinovirus. Yeah, there are other viruses as well, but those viruses you will not be able to see. But if you want to see bacteria, you're going to see them in any case because in the mouth there are so many of them anyway. Huh? So that's a little bit the question: is how quickly is this able to dry? And let me see. Let me spread this carefully here as well. Okay, to give it a little more surface area. If you give it more surface area, then it might dry a little bit faster. But then again, I don't know. Yeah, we're gonna see. What we could try to do is we could just try to stain it with crystal violet, because in this case they're definitely going to stain blue, whether they're gram-positive or gram-negative. Some comments and questions are here again, which I'm going to go through in a minute or so. Okay. Yeah. So let's have a look here. So maybe I'm not gonna do the complete gram stain here. Yeah, see, it's already dry. So I'm going to heat fix this. And always, you see, you always got to be careful. Always move burning stuff away. Just make sure that the camera doesn't. So. Yes, so hot enough. I'm now going to let's have a look under the microscope directly, okay? Let's wait again until it cools a little bit. You know what I'm gonna do? Just a second. Look what I got here. Petri dish for cooling. Yes. So, I'm going to now do the following. I have to move away the scope view. So again, let's put it away. I'm going to remove now the two slides. Let's put them to the side. Here and here. Different magnification. And let's just look at the saliva without anything here. I'm going to now. This is now a little bit of a guesswork. Here, are we able to see something here? You should never do it like the way that I'm doing it here. You always start with the lowest magnification and then work yourself upwards. You always start with the lowest magnification and work yourself upwards. But yeah, see, that's not even the correct one here. So back to the lowest magnification. And here we go. Okay. Yeah. Of course, lots of epithelial cells. So in case you want to know what we see here is all of those things here. These are all cells from my mouth. Bacteria probably scattered in between here, too difficult to see. Okay, so I'm going to now do the following simple stain again using crystal violet. Let's put away the coffee warmer. And now I'm going to do that, what was requested. I'm going to, after every step, I'm going to put it under the microscope. Because, yeah, I've also not tried this, my saliva, before. So this is actually also for me something new. So number one, and a toothpick to spread it because I want to save the stain. Okay, here we go. 1 minute. Some comments. Is crystal violet useful for any other kind of specimens? I know safranin is used as a counter stain in several things. Honestly, crystal violet is extremely powerful, stains pretty much everything. Okay, yeah, I've also used Löffler's solution, which is methylene blue, but I find crystal violet gives you a stronger stain even. Yeah, it's a question of trial and error, I would say. Yeah. Do you smoke? Tobacco smoking affects the salivary gram-positive bacterial population. I don't smoke. Okay, yeah, that's quite well possible. Yeah, that smoking or even the food that you eat and many things actually have an effect on the flora of your mouth. Yeah. Okay, I have an infection. Oh, we talked about that already. Yeah. One of my scopes came with a hematoxylin, sodium iodide, saffron powder, and polychrome. Any uses for the hobbyist? Wow. I mean, these are stains. Hematoxylin stain is commonly used. Saffron powder is actually the one that I used to make my saffron solution. Polychrome, I don't know. Yeah. Yeah. So essentially, I would say it's a question more of trial and error. But if you research a little bit, then you're certainly going to find applications for these. Usually, different stains are suitable for obviously for different specimens, and depends also what you want to make visible, right? So we're gonna do it again. You already know this. Okay. And yeah. Here we go. Yep, and you see that it's yeah, stained blue. I'm going to quickly dry it a little bit, just to remove. Yeah, and let's have a quick look under the microscope again. I'm switching now the scope view, and yeah. look at this. Okay. So very nice. So what we see here, of course, is the following. You see the cells here in the center, the dark nucleus, and the stuff here seems to be mucus that's stained in the back here, right? But you see, yeah, you've got the individual cells here, yeah, and yeah. What I want to do now is I've not tried this yet. I'm just going to, indeed, I'm just going to move on now. I'm not going to interrupt myself now. I just want to complete the gram staining very quickly because I would like to know if it actually de-stains or not. So what I have to do next is, according to the protocol, I have to add some iodine. So I'm going to do this very quick now. Okay. Let's see if yeah, how this actually works. Okay. So some iodine for 1 minute. Okay. So let's see how this works. This actually, this would be actually, if this actually were to work now, if I'm able to see now some gram-positive cells, bacteria in here, and then that would indeed be a very nice demonstration to see how you're able to make the bacteria visible and not the body cells, right? Essentially, this was the original intention of Gram back in the 19th century, yeah, to selectively stain the bacteria so that you can see them better compared to the remaining body cells. Okay, I guess the minute should already be over. Okay, so what I'm doing therefore next is I'm going to rinse it again. Okay, we can see again, just like before, it's not blue anymore, but it's a little bit dark gray, black. Okay. So I still want to check it now because I'm just curious how this looks like. Indeed, I'd like to know. Look, yeah, the iodine made everything, turned everything from blue to black. Okay. So, and now we're going to start the de-staining by adding the alcohol. I have to always forget to change the camera. So let's do the same thing again here. Let's try to de-stain. You can actually see now that it's turning, blue pigment is coming out. So, yeah, I think this is gonna be a separate video where I just briefly show the steps, because I can imagine that there might be some folks out there who would like to see this. So I'm going to leave it like this. Yeah, I'm going to quickly rinse again with water. I should have held it differently. Okay. So, yeah. Yeah, if the Q&A session is a little bit later, I'm sure you don't mind, because I myself would like to actually now know. Okay, so this is now the de-stained one. Where is the scope view? And let's have a look. Ha! What is this? Where is everything? Ah, look. Ah, de-staining not quite complete yet, but it's actually also quite nice. Do you actually see over here the epithelial cells? You see that they're still dark in the middle, right? Here in the center, but bright on the outside. So I have not waited long enough yet. Okay, I've not waited long enough yet. So what I'm going to do is I have to, I'm going to give it another alcohol rinse, right? See the epithelial cells here are turning lighter and brighter. My body cells are losing the color, but not in the center yet. Yeah, so this means I have to add a little bit more alcohol. This was actually quite an interesting observation, because it actually shows that by doing a little bit of a you can do a microscopy quality check here. So I'm going to add again some alcohol here, and maybe I'm just going to wait a little bit for the alcohol to do its work. Okay. Okay. Let's see how well this works. So I'm going to give it maybe a minute indeed to allow the, but there's nothing coming out anymore. Okay, nothing coming out anymore. Yeah, I think it should have completely de-stained by now. Next thing is the following. Yeah, it says I should quickly rinse again to remove the alcohol. And the last stain is the counter stain, would be now the safranin. So let's give it also a minute. And then basically we'll know the answer on whether body cells are able to keep the crystal violet stain or not. Okay. Okay. I will, before I do the following, is the mucus protein based? Yeah, I would say mucus in nature very often does contain protein or polysaccharides. In the case of human mucus, I don't know. But a lot of slime and mucus actually are long chains of polysaccharides. So if you have anything kind of long chains, be it protein, like for example egg, right? It's this kind of mucousy, but it's protein, right? But also polysaccharides, they have these long chains and they kind of increase the viscosity and basically make things slimy and things like that. So I just basically declare the minute over. And I'm just also going to yeah, rinse this off. Okay. I'm going to now dry it again on the coffee warmer. And the hope is, and that's the hope, and again, this is a demonstration effect. Yeah, the hope is that we're able to see some gram-positive bacteria in my mouth, and that the cells, the body cells, that they stained red. So I'm going to put it again on the, here on the coffee warmer, and yeah, let's give it a minute or so here. Oh, thank you very much for the donation. Thank you very much. I just see that. Yeah, thank you very much. Yeah, the saffron on the paper looks like blood. Yes, indeed. Also here, it's almost like you poked yourself. Yeah. So, how do you dispose of the waste fluid? So this one over here. I basically do the following. I just dilute it with water and I pour it down the drain. Not a problem. These stains here are not toxic or anything, especially not at these concentrations. The alcohol is not a problem. The iodine is not a problem. However, I always dilute it first. Otherwise, it's going to stain the washing basin. You know and then you won't get it out there. Yeah, but these stains are actually not a problem. Okay. So, yeah, I just see that it's slowly starting to completely dry up now again. And yes, it's finished now. I'm going to quickly cool it here again. Yeah. And yeah, then this is going to be the actual real test of whether it worked or not. When I worked in the lab, I sometimes had to do these stainings and all the experiments over and over and over again, several times. Simply in the sciences, you always want to be able to repeat an experiment. Yeah. So I'm going to do the following. I'm going to lower this here again. I'm going to go back to the scope view. Here is the scope view. I'm going to lower it again here. This one goes in. Oil drop goes on top. And careful, careful, careful. Always use the oil immersion lens. Don't ever forget about that. Yeah? Otherwise you're gonna end up contaminating or messing up your other lenses. And I'm now raising it all the way to the top again, because remember, the stage remembered. I have to go up with the light. I'm going to move this back and forth a little bit to spread the oil. I'll try to find the focus. Okay, let's increase the brightness here a little bit. Okay, and I don't know where. Yeah. Very nice. I'm happy. I am happy. I'm happy. Why am I so happy? Why am I so happy? Because it worked. So I'm gonna explain. Beautiful. This is actually better than I thought. Here it did not completely destain that here. Look. Very nice. Okay, I need to explain this to you. This is actually quite happy. So those red patches that you see here, they seem to be the cells of my mouth, but those dark ones over here, do you see those? Bacteria. Okay, over here especially, you can actually see them, that they, the gram-positive ones, they stain darker, and now we're able to see them, and we were not able to see them before, right? So let's have a look here. Yeah, definitely. Let's see. Let's go to a place where we have. Yeah, these here are those red patches are the epithelial cells here. Here, this one did not completely destain the black. Yeah, but look, the bacteria here, right? Rod-shaped bacteria. Yeah, they are now nicely visible. So these are all bacteria of my mouth. And definitely. But where are, let's put, see these are all cells that did not complete, now these are the cells, they did not completely destain. I wonder what these patches are, because they seem to be actually they seem to be here actually the cells, because they're from the size. Look at this. Are these all bacteria in the cell here? Yeah, they're kind of sticking to the epithelial cell. Yummy. It kind of shows how many bacteria there are in your mouth, or in my mouth in this case. Yeah. Let me see where else can I, maybe let's move over here a little bit. Yeah, these here are not completely destained yet. Ah, yeah. Here again, that's also one of those epithelial cells with a nucleus in the center. Okay. And those things here, all bacteria. So that's actually, yeah. And we could not see them before. I don't know. Look, could this be? I don't know. It's a question of interpretation. Could this bacterium be a little bit red? Then it would be gram negative, right? Yeah, so some of them are, yeah, and this one seems to be gram positive down here because it's stains dark blue. Ah, definitely. Look, this one over here, a long rod-shaped gram negative bacterium over here, because it stains red. They're here, they're blue. Yeah. Okay. So off topic a bit. If you separated out a single bacterial cell, how long could you potentially keep that one cell alive for ignoring any new cells it splits? So basically, how long could you keep it alive? Well, honestly, if the cell forms spores, endospores, which are basically stages where the cell is resistant, you can keep it alive for hundreds, if not thousands of years. Endospores, which some gram-positive bacteria form, can be incredibly resistant. Otherwise, how long could I keep it alive? Well, I guess it depends mostly on the environmental conditions. This one is quite nice. Look. Can it really be like that, that around the body cell there are so many bacteria? Interesting. Yeah. I would say that this depends mostly on the environmental conditions, right? But I know that bacteria have been kept alive for a very long time. Yeah. So, you know what, people? I'm not going to, I did promise to talk about some of those questions here, so I will do that as well. So I'm going to just, I've got a whole bunch of questions here. And because I already answered a few of them, I'm just going to do this fairly quickly. Okay. Any risks? I talked about that. I talked about this. Yeah, I bought my first microscope recently and it came with 185 objectives. These are the short barrel objectives. Many traditional microscopes also, for example, my old Olympus microscopes also had a 185. I was wondering if I can exchange them for 195 objectives. Only if you have enough space. This means if you're able to lower the stage sufficiently, yeah, that the larger objectives fit. If you're able to do that, then it should be possible. Okay, yeah, the question is if it's worth it or not. Yeah, so it depends really a lot on the space of the stage of whether you're able to fit in the larger objectives. Yeah. I talked about this. Can I stain? Yeah. I wish my instructors were as knowledgeable as you when I did. Okay, well, thank you very much for the compliment. Okay. Is multiplication or time sometimes used as an identification criterion? Generally not. I talked about this as well. This is biosafety level 2. Interesting, we use a cover slip to spread out and then heat fix. Yeah, you can of course also use a cover glass. The way that I've learned it is to directly actually, to not use a cover slip and to directly put the oil on the heat fixed bacteria. Yeah? Both seems to work. Yeah, I need to put on my reading glasses. DNA, I talked about this one. From what I've read online, modern procedures recommend not testing the heat on your skin due to the fact that idiots have badly burnt themselves doing this. Oh my gosh, yes. Yeah. Yeah, this if you just joined, this was kind of the test of whether the slide was sufficiently hot or not. Yeah. But back to serious. What I've actually done in the lab, when I worked in the lab, what I've done is when I used the gas burner, I did not even heat test it anymore, the slide, but I simply had a time that I pulled it through the flame in a certain speed 2 times, and then I knew that it was hot enough, but not too hot. Yeah. So I actually then later on in the lab I did not test it anymore, but I simply had my procedure, yeah, and a certain speed of pulling it through the gas flame, and then there was no need for actually testing it. Okay. Yeah, and more, but doing it like a blood smear. I've read that crystal violet can be substituted by methylene blue for gram staining. Might be interesting. Okay, yeah. Just to let you know, I'm setting an alarm. Okay. Okay. In astronomy, yeah, there was actually a partial solar eclipse today here in Europe. Unfortunately, due to the rain, I could not see anything. But actually, we did have a partial solar eclipse today. So a mixed bacterium group will show red and blue. Yes, then this is actually what I also was able to show you, that you're actually able to then see different bacteria on the same slide. Okay. Yeah, a Beerman funnel setup. Very, very nice. If you've got pictures, please do send them. A Beerman funnel allows you to collect, for example, water bears, rotifers from a sample. Yeah. Okay. I'm just quickly going through the questions here. I wonder if you could partner with someone somewhere who would be able to get a flu vaccine shot to show under the microscope. Honestly, a flu vaccine shot is not going to show you a lot because the antigens that are used, yeah, they are too small to be actually seen with a microscope. So vaccines are individual antigens or maybe even whole viruses, but even then you're not able to see them because they're below the resolution limit of a light microscope. So that's unfortunately not possible. I know several people already wanted to see viruses, but I cannot do that with a light microscope. Yeah. Oil immersion and phase contrast I would not mix. You can try it out, but I would not mix it because phase contrast, then you're losing the how do you call it, the color. Yeah. Saliva, I just did that. Yeah. Would epithelial cells color and bacteria differently? And the answer is a clear yes, because this is what we've just done here, right? So what we can see here is that's the epithelial cell. That's the nucleus in here, which is still stained blue. And these things out here, yeah, they look very much like bacteria. In this case, gram-positive bacteria. Okay, the gram-negative ones, the red ones, probably are also there. I've seen a few of them, but they have a lower contrast, and therefore you don't see them quite as well. So what I'm going to do now, what time is it? It's already 1.5 hours. I'm going to quickly go through some remaining comments or questions. So Hadrian is saying, to repeat your experiments, can you use methylene blue, aniline blue, or astra blue as a substitute for crystal violet? Iodine and saffron are available. Short answer, I don't know. The only thing that I do know is that the crystal violet reacts with the iodine to make a crystal violet iodine complex inside the cell, and I don't know to what extent the other stains are able to do that as well. Okay, but however, general answer, and this is how also Gram and other scientists have figured it out, they just tried it. This was a purely trial and error. So back in the day, they obtained dyes and stains, and they simply tried it out. I think that's also something that I would recommend, and that's what I'm doing as well. We just tried, right? And some things might work, and some things might not work, and then maybe we discover a few new things this way. I quickly skip only if things are for me. I understand PCR is used to make multiple copies of DNA, but how do scientists determine the sequence of a DNA strand? That's the so-called standard Sanger's dideoxy sequencing method. Could you explain the process briefly? This is not a brief explanation. This is actually how sequencing happens. Is a variation of PCR, and I cannot explain this briefly, but you use basically gel electrophoresis for that, and there is no easy way for me to explain this, but very briefly, sequencing nowadays is done by machines. So basically you give the original DNA, and you do the reaction, and then the machine will sequence them, and then you have gel electrophoresis, and you're able to read off the base sequences. So this is actually a standardized procedure, but I would say I need to make a drawing in order to explain this. Okay, and it's called the Sanger's dideoxy sequencing method. That's basically allows you to determine the base sequence. It's a pretty fancy and cool technique. Yeah, have to go now as well. Okay, and I think I also have to go. Okay, the bacterial cluster around the epithelial cells. Yeah, I saw this with my own cells and methylene blue as well. Interesting. Yeah. I don't know if they have a specific affinity for the cells or not. Yeah. But first of all, I would like to thank you again for your Trinova Labs for the financial contribution. I'm using the money always to buy stains and microscope slides and so on. And I'm going to show you for these live streams and to make videos. So thank you very much. And I'm going to actually call it a day for now. Because 1.5 hours of talking actually is a long time, and some of you are actually, probably in the middle of the night somewhere. Yeah, I'm going to call it, also a day today. I hope that you like the video again. I'm going to think of another topic for next week's live stream. Suggestions are always highly welcome, and of course I will. And the micromanipulator, still have to work on that. But maybe something I would like to show you, because I do have a gas burner right now. It is possible to make very small pipettes using glass, and maybe I can actually demonstrate that as well, so that you can actually pick up individual cells under the microscope even without a micromanipulator. Okay, so we're going to have a look at this possibly as well. I want to thank you that you joined in. I want to wish you a very nice day again. Have a nice weekend. Hope to see you again in an upcoming live stream. And happy microbe hunting. That's always the greeting that I say. Bye-bye and see you around.